without further ado, Christof.
Thank you, Gabriel. Just to give you a motivation, I don't think you need any, but it's a simple computation. So we've got roughly 6 million cones that we use in this sort of light outside in the bright sunlight. Right now, the rod, they can be modulated roughly at 20 to 30 hertz, call it 25 hertz. Signal to noise is roughly 100, so call it 7 bits, 128. So that gives rise to just flowing through my retina as you look on your screen and look around the world, 10 to the 9 bits of second of visual information per each and every second streaming up into your brain. There are probably at least we know there are at least 24 different target areas that are targeted by retinal ganglion cells, and some of those include the LGN and the V1 that the course here focuses on. Now, if you look at the output end, whether it's typing on our keyboards, people have done this quite well, done lots of interesting psychophysics here, or whether it's speaking, or whether it's reading, it's roughly 50 words a minute typing. So if you look at the entropy, etc., you can generate 10 bits per second of motor information, let's say on your fingers, or whether you speak roughly 10 bits a second. So you take in 10 to the 9 bits and you can produce appropriate output, because of course this is not high entropic output, but it's actually meaningful output. And we know people have done some psychophysics on it. You can do this totally. It requires visual attention, but doesn't even require awareness. You can do it so fast that that conscious awareness of what you type typically comes several hundred milliseconds after you actually typed it. There's some cute experiments to show that. And of course, the same visual motor system can do all sorts of other things at an instant. You can, within seconds, switch to a bike, eat with a fork, or recognize Jennifer Aniston and others. And so the question that here we are tasked to trying to answer, how does the brain do it? The brain roughly has 10 to the 11 neurons. They fire at a couple of hertz, so you got roughly 10 to the 12 action potentials each and every second. And somehow out of all of that furious activity emerges behavior, and then also emerges, I'll talk about it tonight, this obviously gold and white dress. Right? And anybody who sees it otherwise is, well, different from me. But this, I do want to remind you, although I don't think you're going to talk about it at all, is that this brain of ours, when we look at things, we cannot only do recognition, like for example Jim demonstrated this morning, using CNN. We cannot only say recognize it's a face, or it's my dog, or it's a German Shepherd dog, but we actually have a picture in our head. We actually have conscious experience that parallels or that follows the behavior. So the question, how does the brain do it? All right, so the human brain, you all know, this is still the best known atlas of the brain, was done roughly 100 years ago in the monkey, in the rat, and in the human. And so Corbinian Brodmann, a German neurologist, using purely cytoarchitectonics, in other words, he took Nissl stains, still the standard, and then he looked very fine, and he saw the distinction in these different brain areas of cortex. And in the human, he designated 48 areas, numbered. He stopped, and some he only found in the monkeys. So in the human, they are the Brodmann areas, 1 through 52, based just on structure, tiny differences in the way these neurons and the cell bodies and the myelin is arranged. And that's still by far the most popular map we use today. We just have an update coming out next week. It's a spectacular, entire special issue of Journal Computational Neurology dedicated to a very high resolution atlas of the human brain, where we have PV stains, and we have neurofilament protein antibody stains together with Nissl. And so now, 100 years later, we can distinguish 750 different gray matter areas in the human brain, 750. Here at 52, and several hundred white matter tracts. So, the number of areas, at least the structural, has sort of increased by a factor 10. And then you also saw, this came out a couple of months ago in Nature, the David van Essen human connectome, that using a variety of functional measures, particular resting state activity and correlated with sensory-driven activity, they now have in cortex alone, so the 750 structures in the entire human brain. It's an upper bound, that's a lower bound, and here now, based on functional criteria, 180 structures. The brain is similar to what we experience as neuroscientists, is similar to what happens in astronomy over the last several hundred years, last 5 centuries in fact, that each new generation of astronomers, as their instrument becomes better, realize that the universe is a bigger place than they used to think. So of course we used to all believe that everything turned around the Earth, and then we discovered, well, that's not quite the case. There are other planets that together with us rotate around the sun. Then we realized the stars out there are actually sun like ours, and we realized we live in a galaxy, and then at the beginning of the 20th century we realized there are thousands of galaxies. So now it turns out there are roughly 10 to the 12 stars in each galaxy, in 10 to the 12 galaxies. So we live in a universe that, in the observable universe, that has at least a mole of stars, 10 to the 23. It's quite an impressive number. Vastly more stars than there are neurons and synapses in your brain, incidentally. And now, of course, string theories in their reckless abandon, they postulate an infinite number of other universes with whom we don't causally interact, but they do exist. So, and same thing in neuroscience. As our tools become better, we see more and more complexity. So now we have 180 functional areas, 750 structural areas. Some numbers that I always taught at Caltech, because I think these numbers are really crucial to remember. So it's 1.5 kilo, right, our human brain. 86 billion, so this comes from the lab of Herculano Husserl, the roughly 86 billion neurons, and same number of glia and astrocytes and non-neurons. So it's simply not true. There is this urban legend that goes around that we have 10 times more neurons, that our ratio of non-neural to neuronal is 10 times. That's simply not true. An accurate count says they're roughly the same number. They have differential distribution. In the cerebellum, there are more. In cortex, there are less. But roughly, there are as many non-neuronal cells as there are as neurons, 86 billion. Now, if you just focus on cortex, the one we believe relates closest to intelligence, you've got 16 billion cortical neurons. So that's roughly one out of every 5 neurons. The vast majority of your neurons are, in fact, in your cerebellum. So roughly 3 out of 4 neurons are in your back of the brain, the granule cells in the cerebellum. Everything else that people care about, it's another interesting number, is under 1%. So all these other structures, the amygdala and the substantia innominata and the basal ganglion, the diencephalon, the brainstem, some of which of course are absolutely crucial. You would be in coma if you didn't have them, is under 1%. Now interesting, this is a challenge for those of us who believe that we are at the top of the food pyramid and that we are the most intelligent, and that intelligence relates to cortex, because you have an animal here, a pilot whale, it's actually a dolphin, whose cortex, not whose brain. Of course, we know since hundreds of years that other animals like elephants and whales have bigger brains, but this actually has a bigger cortex, twice as big as ours. Not clear what it does with all that cortex. There it is. It's a lot of cortex. So it's really a physical sheet. The best way to think about it is like a piece of cloth, 2 to 3 millimeter thick. It's really the size of a pizza, roughly 1,200 square centimeters, so it's like a 12-inch pizza. And you've got 2 of them, and of course they're highly wrapped up and highly convolved. They're roughly under each, yes? No, it's still lower. It's still lower. It's lower than us, but it's cortex. So it is not clear why you should have, while if you have a bigger, so this argument has never made sense to me. It makes sense, okay, for motor cortex, I have more body, I have more muscle, presumably, because they're limited in size, so therefore I need a bigger motor cortex. It's not clear to me why I would need a bigger V1 or bigger dorsolateral prefrontal cortex, because my brain size is bigger. So it's actually numbers of neurons. It's not just size, because of course we also know as across species the size of the cells. So for example, in a mouse, everything is smaller than in a human. Nobody's recorded. I don't know what the size, but here they actually counted the number of cells. So it can't be really discounted by using an allometric argument. It's a big puzzle. There are roughly 100,000 neurons under each square millimeter. In a mouse, it's 50,000. In a cat, it's 80,000. I don't know exactly what it is in a monkey, but it's roughly on the order of 100,000 cells. So that's a couple of 100 trillion synapses. Now the challenge with this is, and some of you together with me just came over from that debate. These neurons are not a homogeneous soup, so you can't just treat them like a Hopfield network or general. If you want to be very true to the biology, you can't treat them just as a homogeneous set of neurons, and all you have a Wij, because as I'll show in a second, they're probably on the order of 100 different cell types. And this is a challenge because our models, our mathematical models, computational neuroscience models, typically have highly homogeneous population. But if we know anything about the retina or about other structures of the brain, they have highly, highly heterogeneous cell population with distinct function, with distinct projections, with distinct gene expression. It's a challenge. I'll come back to that. And of course, to remind you that it's a physical thing, it's a physical tissue, physics matter. Here you see a vascularization in a monkey. From a monkey, people have done the same in humans. So this is if you plasticize the arteries and the capillaries and the veins and remove all the tissue, this is what remains, right? So this is roughly 1.5 millimeters of cortex. And we need this, presumably because we need to get power. We need to get oxygen to the cells. And of course, the change in oxygen in response to metabolic demand is what drives fMRI. But it really brings home the physical nature of this computational tissue. It's a tissue, it's really a sheet because the width is relatively constant across evolution. It changed maybe from 700 micrometer to maybe 3 millimeter or something. So this change only changed by factor 2 to 4, while the width, the size of the cortical sheet across evolution, mammalian evolution, changed by a factor of 50,000 or 90,000. So it's really, and the predominant organization that hasn't changed really. So think of it like a forest. You have this predominant organization, but then as the animal evolves, you get different species become smaller or larger. And it's a sheet, and if you ask where does the sheet end, does somebody know where the sheet tucks in underneath? What structure is there? What? Hippocampus. All right, so here you have a classical picture from David Tubal's book, so you've all seen this. So I think of it as a planar 2 plus epsilon dimension. Computational tissue varies factor 100,000. It's relatively uniform, the emphasis on relative. And so the question is, that has excised theoreticians since ages, is what is the canonical columnar operation that goes on across cortex that makes intelligent so robust in us and in related species? Talking about the evolution, this is the oldest mammal, at least the oldest mammal for which we have evidence for, a skull. This is a tiny, like a mouse-like, rodent-like animal. Hydrocodium was found in China, and they did MRI scans in a beautiful science paper. You can see the earliest cortex was olfactory cortex because the ecological niche 200 million years ago was at night, right? The only way mammals could survive, I mean, look at the paper clip. It's a tiny mammal, right? The only way we could survive is at night, and we needed vision wasn't terribly important in nocturnal creatures, so smell was really important. And it looks like the oldest the oldest cortical structure is aliocortex, is olfactory cortex, and then it developed from there. Probably first touch and other proximal senses, and vision came later. Vision that we and you guys put such and of course we as humans put such a bias on is a later arrival.
How can they infer the shape of the brain from only having the skull?
No, so they infer just from the endoplast. This is inferred. The pink stuff is inferred. Yeah, correct. All right. So now let's zoom in. So this is a Nissl stain. So here you see each of these. Think of it, each of these sort of is a Nissl body, and you can sort of click on it, and then you can in principle expand it if you do the Golgi or biocytin or fluorescent stains, and you get these gorgeous neurons and their axons. Now, if you look closely, as I mentioned, you see they're not only, in fact, here you can see this, right? So this is a tattoo from when I went to the Allen Institute. I thought I should get committed to the mouse, and so I got a mouse tattoo here. This is TD tomato. Anyhow, so here you can see different cell types. This is from a picture from 1890 or so. So you have different cell types. So it's an old. Anatomists know about it since 150 years. It's only very recently percolated through to theoreticians that there are different cell types, and that you cannot just have a single W. And you can't just sort of treat the brain with a single WIJ. You have to we have to start thinking about why are these different cells and what's their function. We know this best in the retina. So this is from Dick Maslin, where we now have a semi-complete account of all the different cell types, partly because we can they have beautiful precise positioning, the layers within the interplexiform layer. So the axons sort of ramify very specific layers inside the retina, pretty much across all in most vertebrate and certainly in mammals. And so that allows us to align, and now we're beginning to get genetic identification and genetic handle on, although here it's mainly based on anatomy and function. So we know they're cones and horizontal cells and bipolar and amacrines and ganglion cells. And ganglion cells, there are roughly now 24, and they project to different places. Here we can identify somewhat the function of them, because some are direction selective, some are not, some are luminance detectors, and as I said, they project to different places, although it's still a mystery why we need so many. But here we have a reasonable handle on structure-function relationship in the retina. And so nothing in the retina makes sense without referring to what cell type. Nobody would publish a paper and say, well, I recorded from 20% of all of cells in the retina, and they do this, without saying I recorded from this particular ganglion cell class or that particular amacrine class. And we also know, for example, when we're beginning to understand when you're looking at pathologies, you know, when we're looking at pathologies of the brain, which of course if you think what's the reason why the brain is so popular and the brain is funded by so many, there's brain projects and there's China brain project, etc. It's not because people care about making abstract models about the brain, but they want to get cured from their diseases, or they don't want to get the disease in the first place. And so here, for example, we know there's a particular type of pathologies where children, they make these nystagmus permanently. They can't stop themselves. And we now know there's a beautiful mouse model. There's a paper in Neuron by Botha and Roska in Basel. We now know that's due to a specific type of defect in a specific type of synapse from an established amacrine cell to a particular type of retinal ganglion cell. And I suspect that many pathologies like schizophrenia, etc., for which we know there are thousands of genetic variants, is probably due to a very specific perturbation in a specific gene whose proteins are expressed in a particular cell type. So it's really ultimately, certainly to cure diseases, this knowledge will be important. So, we know since ages there are different, of course, spiny, non-spiny, and there are all these different inhibitory cells. I'm not going to bother you with them here. This is the latest from Henry Markram. He's done this very careful comparison in rat somatosensory area. There are at least 55 different in one cortical region based on morphological criteria. And there are 12 different electrophysiological, and they identified 218 using 10 years of research, very careful experimentally driven research. They identified 200 different morphological times electrical type in one particular area. They don't know, of course, the vast majority. We have no idea what they do. We just know if we look at the different, where they project to and whether they have a bushy dendritic tree or an extended one, or they go like this, there seem to be roughly 200 different types. If you cross that with the different types of electrical behavior, do they respond in a step? Do they not adapt? Do they adapt? Do they sputter? Do they burst, etc.? So it's a challenge. And we don't know to what extent these cell types are really in cortex are really functional. It's very difficult to know right now, because for the most type, still we don't have transgenic animals, right? So because it would be cool if for every cell type you have a specific transgenic animal that you can then target, you can express that, you can turn that neuron on, you can turn it off with halo and with channel rhodopsin, and you can image it using calcium imaging. Then we could maybe better understand why we have these different cell types. So this is from our latest paper. So if we do single cell transcription analysis, we look at it in thousands of cells, do RNA-seq at the single cell level. We get, in this case for mouse V1, we get 42 different cell types. And that matches roughly well up with the cell type ontology that's derived based on development. So, the answer may not necessarily be 42, but it's going to be in that order of magnitude. Now, most of what we do at the Allen is we're looking at two types of tissue, human tissue. I won't talk about that at all today, from neurosurgical tissue that we get once a week from neurosurgical operation, as well as the common laboratory mouse, C57J, black. Okay, that's our animal. So that's the size. That's a human brain, one of our human brains, and that's a mouse brain. It's pretty much a factor of 10 linear dimension, factor 1,000 in volume, roughly. It's quite similar. Of course, you're all expert in neuroanatomists now, and you can tell me which one is human and which one is mouse. Which one is human?
Left.
Who votes for left and who votes for right?
Left.
Right, yeah. Yeah, it is. It's 3 times bigger, so we had to scale it down. So the neurons appear smaller. They're actually bigger than over there. But it's, as I said, it's remarkable similar, and in terms of the type of neurons expressed. There are myriads of differences, but overall it's remarkable similar. In fact, only if I give you, a little grain of matter, and I say, well, this is 1 by 1 by 1 millimeter of cortex, it would take an expert armed with a microscope to say it's a monkey or human or mouse or elephant, right? The basic stuff is really very similar. So here's the comparison. It's roughly 1,000, pretty much. So it's 68 billion, 71 million. Cortex is 16 billion versus 14 million. We have many fewer visual. Well, here now this is all. We know we're 180. Here the current estimated 30, but it's also creeping upwards as people get better techniques. And we can train animals. So this is something Sean Olsen at the Institute did. It's on a running wheel. You must have seen things like this. So here it recognizes, particular pictures. This is me with the count. It's our patron saint, the count.
Question for you on Nature Transcriptome paper. Are those also done on C57 or are those across strains?
It's all C57. It's all adult.
Is there potential that like in an inbred strain?
Oh, we know already they're different. We've done that comparison. Different strains have different expression. So we have the in situ for that to show that they differ. Sometimes they differ quite dramatically, different strains. And quite different. If you've ever seen a mouse in the wild, right, they're very different. So this is one worry of many I have, that we study, and most people study mice that are inbred since hundreds of generations. The first C57 was developed in 1921. They haven't seen the outside of our world since 90 years. And they only have sex with each other, right? So if you think about that in human terms, you take a population, you isolate them for hundreds of years in a very particular environment, in a lab environment, you wonder what you're going to get. I'm just saying. All right, why is studying brains so difficult? So, neuroscience is now 120 years old, 150 years, but we've made very slow progress, particularly when we think about understanding whole systems, including very simple system like C. elegans with 302 neurons, or curing diseases. So the challenge is, and this needs to be reflected ultimately in new types of mathematics. So we all know brains have large number, but they're highly heterogeneous cell types. On the order, we're pretty sure now 1,000, maybe 500, maybe 2,000. In fact, there are now a number of projects that are getting ready, including us, to do cell type across the entire cortex and across the entire brain, both in mouse and in human. But it's going to be 1,000 or several thousands. Interacting, each one of them interacts in very complicated but apparently very specific patterns with 10 to the 3, 10 to the 4 other cells across a spectrum of scales, right? From milliseconds to, of course, our lifetime, and from micrometers to tens of centimeters. We don't have a list of the cellular components. It's difficult. So if you think about brain TV, when we have, take 10 billion neurons, that's a TV screen of 10,000 by 10,000 pixels. You're trying to understand what goes on in the brain, but you can only observe. Think about a movie. Think about a movie on a screen where you have 10,000 by 10,000 pixels. But the only thing you can see is either something very fuzzy, if you blur your eyes, you can sort of see, fMRI activity, or you can see very high resolution, but that's like, 0.0001 of the pixels. And now you're trying to understand the story of the underlying movie that's ongoing, right, from these 100 pixels, or 1,000 at best right now. We have very few, unlike other fields like molecular biology, let alone astronomy, we have very few accepted standards for relevant phenomena. So even spikes, you think a spike is a spike, it's a spike, but then you talk to a 2-photon calcium imaging person, you talk to somebody who patches in slice, you talk to somebody who's extracellular recordings, they all have different definitions of spikes and different algorithms. So that's a problem. There are no central unifying projects. And they're roughly on the order, if you estimate the population of neuroscientists, there's 60,000 neuroscientists. Roughly, there's an order of 10 to the 4 different labs. And they all have different questions, methods, protocol. And of course, by design, the system is to be orthogonal to each other, because you get a PhD only if you do something that's significant different from what your predecessor did. And your advisor, she only gets a grant if she proposes something that's significant different, because if it's just survey, people will say, well, that's just fishing. That's not hypothesis driven, and your R01, your committee will kill you for that. And so everything is designed to do, because we want to do discovery, right? And so everybody does something different, which is a problem if you want to understand a few things in great, great detail. Plus, of course, we have the replication crisis. And so universities are great, fantastic. I spent 4 years at MIT and 27 years at Caltech. They're fantastic, but they're not set up for certain large-scale systematic efforts. And I'll get that. So how do we approach this problem? So the argument is that in addition to do the traditional small lab PI-driven approach, where the supreme outcome is a series of papers in high-impact journals, that system that's fantastic for discovery, we need to supplement with a different system that sort of I would characterize as big science and team science and open science. And I'll talk to you about what I mean by big science, team science, and open science. So let's switch to us. So who are we? We're an independent, non-profit medical research organization. We were started 2,000 years ago by Paul Allen. We're growing rapidly. We're now 300. We'll be 400 in a couple of years from now. The culture is it's closer to LHC, for those of you from physics or LIGO, than it is to a traditional university lab. It's somewhere between a university and a biotech startup. So we are focused on a handful of larger projects that can be done at scale. We don't want to compete with university-driven PI research. We want to do things that can't be done at a university, and that require things that we can do systematically across many years in a very highly reproducible manner that require tight interaction across many different disciplines. So when it was me coming, when I left Caltech and came, we initiated a 10-year program in 2012. For building cellular-level observatories for mice and human cortex. So we focus on cortex, only cortex in mouse, visual cortex, and human middle temporal gyrus. That's roughly our budget. And of course, it's all funded, or 92% of it is funded by the generosity of a single individual, Paul Allen. And I'm not going to belabor this. One of the big differences is between the way you guys all do your research and we do. We have 10-year plans. So we have milestones. No, I mean, I never had this before at Caltech. Right? You do opportunity-driven research, and I have some personal interest, and I try to pursue them. But here, there's systematic plans certain things that we try to accomplish. We have external advisory boards, and we have product releases. So those triangles are big new products, and we introduce, and then every 3 months we push out products, and we've done that since 13 years. And we just Paul last year gave us a gorgeous new building just next South Lake Union, if you know the area where Amazon is headquartered and where Google is now, and a lot of other tech, the Hutches, a lot of other tech space. So we have this gorgeous glass building now. All right, so we are both big science, team science, open science. And the 3 things that makes us most different, from the get-go, all of our data is publicly and freely accessible. So you can right now, as I speak, you can go to brain-map.org, and you can download every data. Any data I talk to. You don't need an IP. You don't need an IP agreement. There's no login, no registration required. So it's a lot of data. It's curated. So if you have problems, we have a hotline and we answer it. And sometimes we retake data. We have to retake data if there's something funny about the data. And the data typically is accessible. So all these data products, ultimately they end up in either Nature or Neuron, but typically 1 or 2 years after we make the data accessible. And so the project of this focus called MindScope is, if I take this little grain here, okay, that's roughly a millimeter by millimeter. So we are spending a lot of resources and the labor of several hundred scientists, engineers, and technologists to understand in mouse V1, 1 by 1 by 1 millimeter of cortex with every tool that we think today is reproducible. So including EM connectomics, including 2-photon calcium imaging, including high resolution with neural probes, newly developed silicon probes to do electrophysiology, imaging, modeling, and of course also cell types. And so we generate the data for this via a series of pipeline and then put it together into trying to model everything also. So we have these series of pipeline projects, and Lydia's going to talk more about them in detail because they make for great projects. You can download all this data. It's all within a common reference atlas, and you can manipulate and try to begin to understand how the functional data relates to the structural data, relates to the genomic data. So we have a connectivity atlas, a high resolution, several thousand brains now, where we infect specific using viral technology and then fluorescence, and we do automatic serials tomographic reconstruction of these animals, all in the same coordinate system. We now have hundreds, we're going to 1,000 of single cell characterization of these cells, where we characterize them in terms of electrophysiology. We're doing transcriptomic analysis. I'll show you this Brain Observatory data, and we're doing detailed EM-based connectome. This is Clay Reed's effort. Now, why do we study the mouse? Some of you are from human or monkey, and why would we want to study in mouse? Well, the big advantage is really twofold. A, it has a smooth cortex, unlike us. And the biggest reason is really we can intervene in any given point in time now, in time, in space, and in a chosen subpopulation of cell. In principle, this is possible in a monkey, of course, also, but it's much more difficult because it's much more costly, it takes much longer time, and there's a very mature technology, transgenic technology for the mouse that we do our own mice, we build our own transgenic mice that we make available to the community, and so we really begin to understand that quite well. So that's probably the biggest reason. We can just, using genetics, we can just intervene and also label specific cell types and read out their activity. And it's small enough, it's a thousandth of a human brain, yet it is structural overall similar to the human brain. So the claim is if we understand the mouse brain in detail, that'll lead to a better understanding of the human brain. All right, so these are some of this is the visual system, 45,000 retinal ganglion cells, rather than 1 million, projecting to 20 targets, a small subset on the order of 20,000 project to the LGN. There are 18,000 LGN cells, there are 1/3 of a million V1. Overall, there are 2 million in the visual system out of 14 million overall cortical neurons. And Lydia will mention more. Everything we do is with this very high resolution, 300 million pixel, three-dimensional atlas of the mouse brain. Of course, this is another advantage of using transgenic. So we superimposed anatomy of 1,500 transgenic mice, and based on that, of wild-type mice, and based on that, we made this high-resolution atlas. So every time now we record, every time we do an experiment in an animal, or every time we stain something, or we do an in situ, we can always do it with respect to this coordinate system. So we can always return to the spatial location. The mouse visual system, we are not using the mouse visual system because as a model for human vision. We're using, I'll come to you. We are using mouse visual cortex because it has visual behaviors and it's a beautiful piece of cortex, and there are well-developed theories of, like HMAX, there are well-developed theories of vision. Right? So because we know it's quite different. It doesn't have a fovea, mouse, unlike humans and monkeys. It doesn't move its eyes nearly. It can move its eyes, but doesn't move its eyes as a consequence. It doesn't need to move its eyes as much as a mouse. It has two cones, and one of which is shifted into the UV. Yes?
On the imaging techniques that you use, is there anything that takes connections into account?
Yes. I'll come to that.
Okay.
Yeah. Now, it's interesting. Again, we know almost nothing about what this does. So all these papers you see these days published on mouse vision, they use green light here. We know almost nothing about what this system does. but you can measure it. In fact, in the retina, the response to 360 nanometer that we don't see is actually stronger than the response to green light if you measure the retinal evoked potential. All right, so this is using a form of wide field imaging in our mice, so we can identify just like you've seen in the monkey. So here there are 12 areas that we can routinely identify in our mice using intrinsic signal, ISI, intrinsic signal imaging. The biggest, of course, being V1. One huge difference is there isn't that much cortical real estate. Maybe the rest of visual cortex is maybe only 2 or 3 times bigger than V1. We know mice can, there was just a nice study by Chris Neal, we know mice can use their cortex for doing all sorts of visual tasks. Here's a paper from John Mansell that looks at classical psychophysical contrast detection. So you notice, for those of you who know anything about human vision, so our peak is around, in humans, 50 cycles per degree. If you're young, healthy, good eyes, or corrected, we use glasses, you can see 50 cycles, almost almost a minute of arc is our peak frequency. The mouse is far worse. So the mouse is like this in the periphery. So they can see, and if you remove V1 and activate it using optogenetics, they can't do, for example, orientation discrimination, but it's not our vision. Now you got to remember that still means you can do perfectly well lots of tasks. So I used to work on Musca domestica, housefly, and as you know, if you ever observe flies, they can do chases, right? They can sort of try to mate in flies, male chase females. All of that is using visual information, using 30 milliseconds integration interval. So, and of course their receptor field are also huge. They don't have, you can do lots of visual tasks without having high resolution parvocellular system of the sort that we have. All right, so all the data production pipelines at the Institute, it makes it just easier to get common data and always refer to the same animal at roughly the same age and the same strain, because as your question implied, there are different strains of mice and they are different. And of course, mice like they develop and they age, so you want to make sure that you're comparing apples with apples. So we use young adults, so typically 60, 55, 60 days old, both males and females. That's all within a common coordinate framework. And we use common Cree lines that and then we give out to the community, so we always know we're referring to the same transgenic animals and to the same cells. And it all enters a centralized and standardized database. So part of the production effort in structured science is all that data gets sucked into and gets massaged and gets stored and gets referenced. In fact, together with the community, we developed this standard, NWB. You can go to this website, there's a paper out that allows, because we're very interested again in comparing data across labs and across different tools, like if you want to compare EFIS, extracellular EFIS with OFIS, it's very difficult, partly because the data is written in different formats, and you can't even compare it. And so we're using now, together with Janelia and other labs, who are using this standard now, NWB. It's an HDF5-based standard to do easy transposability of data. All right, so this is the whole brain data. Lydia's going to talk more about it. I just wanted to show this is our first set of data based on 400 mice. We now have 2,500 mice. So what this is, you inject a virus in let's say, in one animal in this particular part of visual cortex here. Or you injected a different ABV virus that stains for a subpopulation of cells in this animal in the medulla. And then you wait 2 weeks, and then you sacrifice the animal. So it has a GFP in it that's expressed either in the cytoplasm along the axons, including in the axonal terminals, or you couple it with a synaptophysin that it only expresses in the synaptic terminal. So you can either track or just count the synaptic terminals, and then you can count up, and then you do systematic serial 2-photon tomography with a resolution of a third of a pixel in X and Y, and you do that every 100 micrometers. So you cut the brain into 140 different slices, and then informational you reconstruct all of that. Lydia will talk more about it, and then you get this whole brain connectivity map, which is pretty cool. So now this is a whole brain, if you want WIJ, at the level of populations of neurons. This is not single neurons, this is at the level of population neuron. And the ipsilateral brain, contralateral brain, this above here is the cortex. This would be isocortex. One thing you see, again, is tremendous heterogeneity. We're now doing this in a transgenic specific, in a cell type specific manner. Well, transgenic animal specific manner. This is all wild type. You can see the synaptic weights, if you want, the projection weights. They are log normal distributed, like many things in biology, over 4 orders of magnitude. So you can get, some connections that are 10,000 times stronger than others. Of course, that depends on which sort of normalization you do. So that's pretty cool. So just if you look at V1, the brain area we choose to focus on here in the back. So these are the inputs from all of cortex, just visual cortex into V1, the thalamic input, all the different thalamic areas into V1, all the subcortical non-thalamic areas, and these are the output. The trouble is, if you want to deal with all that data, it's and this is sort of the hallmark of biology, it's very large numbers of very heterogeneous connection. So as I said, some of those will be not all at the same weight. Some have much stronger weights than other weights, but every area, and I don't believe that V1 is particular. It's more connected than any other areas. This is a hallmark of the brain. So we are spending the majority of our efforts, probably every second person or second 2 out of 3 people and $2 out of $3, on trying to get a handle on cell type characterization, both in human and in mouse. And here I'll just talk about the mouse. So one of the things we're doing, we've set up 2 years ago this pipeline called in vitro single cell characterization. So we take slices on a highly reproducible condition of a mouse visual cortex or the human tissue when it comes in, and we put it into our coordinate system, and then we have a standard electrophysiological protocol that's ramps, steps, noise, pink noise, etc. It's a standard paradigm that the modelers devised to probe the electrophysiology. We then have automatic ways to fit the data, both in a variety of GLIF models, of general leaky integrated fire models, from very simple integrated fire to much more complicated ones, with spike-dependent threshold, etc., as well as with very detailed biophysical ones, because we're also injecting biocytin, and we are reconstructing the local dendrites and the local axons, of course only within the slice. And then separate, although we're now moving to doing that in the same cell, we're also doing transcriptomics, where we look what are the genes that are expressed outside the nucleus in terms of mRNA, what mRNA species are expressed inside each cell. So this is done at the single cell level. Typically we have like 8-9 million reads. There are 8,000 different, we look at the 8,000 sort of, we can read out RNA associated with 8,000 genes, and based on that we can do classification. And ultimately what we want to do, of course, we want to combine the classification based on morphology in the same cell with the electrophysiology with their transcriptome. And we're beginning to do that now using a technique called patch-seq. So you can go to our database and go specifically to cell type. And so here what you see is a view of the back of the brain cortex. You see primary visual cortex here. These are higher order visual cortex. And each point here, each disk is a cell. You can click on it. And then, for example, you get this particular description. And I think Lydia is going to do that, is going to show that live. So you can see this is a PV Cre line, so it's an inhibitory cell. This is its behavior. You can zoom in. You can get all the individual traces. So here you can either see them collectively or download each one of them. This is in, for example, the voltage at the cell body, including spiking in response to a variety of different current steps. You can get the FI curve. You get block-phased images. And so here you see the fitting procedure. This is automatically fitting. This is a morphology. So we do the automatic fitting procedure using GLIFs, where we have steepest descent methods that guarantee optimal fit. And then we do using a variety of annealing techniques we got from Idan Segev to also fit a more detailed Hodgkin-Huxley model, either always with active soma but passive dendrites, or in collaboration with Blue Brain with all active dendrites. And you can download all of that. And then, as I said, we are doing this, this transcription analysis where we did this in 1,700 cells. This was over the last several years. And we found these 49 different cells type in V1, of which 42 are neurons, 19 excitatory, 23 inhibitory. And for the most part, there's what you expect. In some cases, in fact, we followed up with morphology to assure ourselves that those are indeed different types of cell that project to different targets, which we do by retro injection and then recovering the cell. And we're now doing this at a much deeper level. We're using more cells, and we're doing this now across other cortical areas in the mouse to try to understand what is how do cell types change? Because if it's true that that cortex is relatively homogeneous, well, if you look in detail, how true is that actually? And, if you look at V1 and you look at some of the higher order visual areas, or you look at somatosensory, or you look at a motor area like ALM, how do they differ from one cell type to from one area to the next? And here the problem is we don't know at this point, so we're using various clustering algorithms. To what extent, if you find clusters, to what extent do these clusters represent real reality, I mean biological discrete nature actually, or to what extent they're just a consequence of the particular clustering algorithm we use? And of course that's a big question relating to all these different cell types. And then the other thing we want to know, how do these cell types relate to the cell types we find in human? So here ultimately we like sort of to get a periodic table, if you want, of the cell types. So we talked about it and was alluded to in the debate over there. So we have this embarras de richesse. Right? So what is the function, if it's true that they're on the order of 100 different cell types computationally, what do they do? And do I need to worry about them in my models? And the answer is, I asked Jim that at the end, at this point we simply don't know. We simply don't know. If you want to be top-down driven, it doesn't seem to be necessary to understand object recognition, for instance, or visual attention. Bottom up, it seems to be, it's very much there in the data. So the question is, to what extent does a final explanation of cortex and its contribution to intelligence requires us to distinguish not only excitatory from inhibitory cells that most people do, but also the fact that there are, that there are different types of inhibition. There are cells, inhibitory cells, that only target other inhibitory cells, and then they project to specific other parts of the brain. To what extent is that critical to keep track of? We don't know. Yeah, the question, is it true? The question that Francis Crick, I think, first asked. So in chemistry, we know that every chemical element can be unambiguously assigned, unless you do nuclear synthesis, can be unambiguously assigned to one out of 92 discrete buckets. Now, is that true also in biology, that every cell that I pick from the brain, let's say from cortex or thalamus or anywhere else, can be unambiguously assigned to one out of 100 buckets? Now, if the answer is, well, there are 10,000, 100,000 cell types, at some point the answer becomes meaningless, right? It doesn't really help you anymore in understanding. So I think it's only interesting if it's a small number, if the number of cell types is much smaller than the number of cells. Or is it true that some types of cells fall on the continuum, which you can also see in some of the transcriptomics data. Like if you look at layer 4, we looked very hard in layer 4. It's very difficult to find discrete clusters in layer 4. But they are different, systematically different if you just sample from top of layer 4 and from bottom of layer 4. So is it possible that there's just a continuum there? We don't know. So that's, at some point we as biologists, we'd like to have some systematic understanding of, cell types and their ontology and how they relate, or, how they relate through development. All right, so then lastly, we built, when I came, one of my dreams was to build an observatory, just like we have astronomical observatory. This is Caltech's TMT 30-meter telescope that's being planned since 5 years, and it may start operating, if they ever get permission, on Mount Kea in another 5 years from now, with a 30-meter effective diameter. So the question is, what astronomy has done very well, to build these large-scale telescopes and to decouple. So there's essentially, people don't do their own observation anymore. They used to do, of course, till early 20th century. They built their own telescopes, like Herschel, who discovered Uranus. They built their own telescope and then operated them. And that's not possible anymore, partly because the cost is just too big for these big, particularly if they're space-based. And also because, as Digital Sloan Sky Survey discovered, what people are now doing in astronomy, at least half of all the papers do that, they do these large-scale survey where you have one set of people that takes the data and puts it out there, and then the astronomers take that data and analyze that data because it's a highly standardized data product. So we are building what we call the Brain Observatory that use three sets of instruments. We use 2-photon calcium imaging, of course. We use wide field imaging, 1P that we can do throughout the entire brain. Think of it like fMRI, but we can do it now on single trials. And we're using high-density electrical recordings, where we sample from hundreds of neurons along a single shaft of highly machined silicon. So this pipeline involves up to 100 people, not 100 people working full-time, but 100 different people, engineers, vets, mechanical engineers, optical engineers, biologists, physicists, computer scientists, data scientists who participate in this. So it's a significant amount of people working over it for 4 years. And you need program manager and all that stuff that sort of that industry has developed. So if conceptually in go various mice, either wild type or various Cre lines that we have that are reporters for where we have GCAM 6, F or S to read out activity in specific layers, layer 2, 3, layer 4, layer 5 of cortex, and then they go through surgery. So there's this entire pipeline where they go through. We have 6 trained neurosurgeons. What they do every day is, operate, so they become highly skillful, and their windows, etc., become highly reproducible. We do intrinsic imaging, so we know exactly where we are. We train the animal behaviorally. We do multiple image sessions where we return day after day to the same location. We do intrinsic imaging again, and then afterwards we sack the animals and we do 2-photon serial reconstruction of those neurons, so we know exactly afterwards what neurons we imaged. So it requires lots of fancy tools. This is our surgery, intrinsic imaging, so lots of different instruments. So that's what we mean by a pipeline. It's not just one tool, it's a whole series of tools. And what's characteristic for these are we have things like SOP, standard operating procedure, where we detail in great, sometimes excruciating detail. We describe exactly, how do we cut things and how do we operate and what sort of tools we use and what criteria do we use for rejection. We reject a lot of our data. Again, all in an attempt to make it very reproducible. We use a lot of fine micromechanics in order to engineer this to make sure we can return to the same place again and again. We use intrinsic imaging. So this is intrinsic imaging in different animals, here in one animal each, having these different Cre lines where we can compute the location of V1 and the other locations. So now if we do 2-photon calcium imaging, we know, are we within V1, are we in AM, are we in ventral posterior, etc. Where are we? And then it took a long time. We figured out what's the standard data set. So now in principle, if you have this tool, you can now, let's first run a standard data set and do it on all the mice, the different transgenic mice in the same place again and again. So we can now capture brain-wise, not just you have one electrode and you go through the tissue until you stop because you hear nicely a neuron that you can nicely drive, like Hewlett and Wiesel did in their days, but you try to do the survey. So what we did for this initial several releases, so we had one release in June, we have another one in October coming up. We're doing drifting gradings, static gradings, sparse noise, spontaneous activity, natural scene from the Van Hatteren, 108 natural scenes, and then we have movie that we show. We show the Orson Welles Touch of Evil. Not because of any, the Touch of Evil is because it has very nice, we here is black and white, so it's a black and white movie, and has long, very long, uninterrupted point of view shots. And so then over the different sessions, so just going one thing and doing it repeatedly, etc., takes on an hour and a half. So we combine them in different ways and do them across different days. So here you can see the data set, and Lydia will talk more about them, how to download them and analyze them. So that's a mouse running, on a wheel behind the touch of evil. It's eye movement with lots of metadata associated with it, and here you can actually see a field of view in V1. So that's a CAX2 line that expresses in layer 2-3 neurons. You can see the activity. So the first release was 18,000 cells, and the vast majority of it we haven't analyzed this data, so the data is all out there. Right? So you can analyze the data as well as we can. We are beginning to analyze it now. Partly we're just limited by having enough people. Just setting up the pipeline and getting the next thing ready is very laborious. So we put the data out there and we'll analyze it. In the meantime, anybody else can analyze it and do interesting discovery. This is, for example, something Michael Bice has done here with Joe Olson, a student of Gabriel, to show that certain. So the nice thing here, what you can do, you can now be much less biased, right? Because you can take thousands of cells across you can compare across different animals, different layers, different areas from thousands of cells, rather than from a very small subset and that's more biased. Here you're biased by transgenic line, of course, right? Depending on which line you have, you're going to see layer 2-3, layer 4. This is all excitatory transgenic line. We're also going to get ready to do. Our goal is to image from every cell population in these areas. So here, for example, if you do decoding either naive Bayes or linear discriminant analysis, where you take account of the correlation structure. So if you do natural scenes, sort of a standard part of the visual stimuli of every psychophysicist, you get very bad decoding performance. But if you go to natural scenes, which after all the animal evolved to see, you get better. In particular, you get much better if you use if you take account of the fact that neurons are correlated, because there's what looks to be a very large degree of variability across trials. So if you look at the individual trials where the animal looks like it does the same thing it runs, you get huge amount of variability. The question is that noise? Is that due to the various other things that the mouse is thinking about or doing, or the position of its tails, or where exactly it moves? That's all, that has to be analyzed. So if you go to the interface, you can see. So each one donates a different experiment in that different part of the brain using a different Cree line. So you can just click on here and get for this particular Cree line the cellular observation based on data there. So that's pretty cool. Now at the same time, what we can do single trials. So think of this like fMRI, but it's on single trial average. It's through the skull. It's so-called CATS imaging. So we image through the skull using GCamp. Well, we can also see the same animal looking at the same stimuli. In this case here, just to give you an impression, at 6 different locations. Now this of course is not. This is cellular and cytoplasmic imaging. And I have a screensaver of this that's plays very long, because it's really tantalizing to look at this activity, and there are all sorts of stuff going on. I mean, look at these big flashes, for instance. Right? See that? What is that? Lots. I mean, we've controlled. It's none of the obvious things that you think it might be. Anyhow, so it's cool because now we can we want to align all the data, which we haven't done yet, right? We want to align the cellular data together with this data, together with EFIS data. I'll show you in a second. That's sort of our goal for the next several years. Right now, we did the 2-photon imaging, and then we did the wide field imaging. Of course, we want to align them. But it sees you the power of this, right? Where you can combine all these different information sources.
Can you recover information about the behavior from looking at these kinds of images? Can you tell if the mouse is like?
Yes, but the behavior here is very simple. The behavior is just running, non-running, and this is here just running or non-running. In a discrimination task, it's again we can recover it, but that's sort of not all that surprising. What? Yes, it is. So we have to do we are doing experiments to try to disambiguate that. And also we want to know where is that response driven by. We, for example, discovered something, there's really cool, there's a structure, my favorite structure in the brain. Who's heard about the claustrum? All right, so this claustrum is if you ask in the human brain based on DTI, what is the most interconnected structure in the human brain? How did you know? It's a claustrum. So when we also done this, so this came out a couple of years ago, we've also done this in the mouse. So in the mouse, the claustrum is a structure that extends from here to here. It's 3.4 millimeter, like this, on both sides. It's connected widely to every cortical area, a little bit less to visual cortex, but certainly also connected to visual cortex and primary auditory cortex, massively to some of the forward and to the hippocampus and perirhinal, massively. And we've done our because we have transgenic, we have a specific Cre line that's only expressed in claustral neurons, and we can see massive signaling. Although it's a tiny structure, it only has 25,000 neurons, it projects massively to all of cortex, and very strong signals, very strong signals. And of course, you might know, so Crick and I published the last paper that Francis Crick wrote, that we think that claustrum is sort of one of the key structures to integrate information as you need in consciousness. Think of it, the analogy we use is like the conductor of the cortical symphony. You have all the individual cortical areas that do their own thing, but you need somebody who can synchronize who can sort of link up and synchronize all of them. And that's what we're trying to test. Anyhow, we'll see. Now, lastly, we're also doing, because these tools, so the advantage of two-photon calcium imaging is you can get genetic identifiable information. Right? You know it's a CAX2 line. And I can send you my transgenic animal, and then you can do imaging, and you know you're recording from the same neurons I am. That's a big advantage. So you see neurons horizontal. The big drawback is it's at 20 or 30 hertz, A. B, you're not seeing spikes, you're using calcium influx, right? And we're doing patching to record the two to precisely understand the relationship. But it's not the same, and it's very slow. So the entire world, again, there are two different worlds here. If you look at the cognitive community dealing with mice, where they do two-photon imaging, versus monkey, because we don't see, mice people don't know anything about gamma and synchrony and all that, because you're just totally blind to it, right? Your two-photon calcium imaging is blind to that. So of course you want to supplement it with high-density recording. So we developed together with it's a consortium with Gernalia. It's really Tim Harris at Gernalia and a silicon design house in Belgium and fabrication called IMAC. These really cool electrodes. We're going to release them to everybody in a year from now, where we have the thing that's relevant for you guys is, so it's a tiny piece of silicon. It's 20 micrometer thin, 50 or 70 micrometer coarse. It's much smaller than existing, particularly the thinness, 20 micrometer. And it has almost 1,000 pixels. They are 20 microns spaced apart in 2 rows or in 4 rows. We're probably going to have 2 separate configurations. Beautiful signal to noise. Noise flows like 3 microvolt or something. And at the base of the electrode, we have electronics for doing filtering and muxing. So we only have 12 wires going out from each probe. And we can select, so it's 10 millimeter long, and we can select 384 out of 966 pixels, the most active one. It's really pretty cool. And there are 2 bands. For each electrode, there's 2 bands. There is a local field potential band, and there's a high frequency band for action potential. So here I'll just show you some data where we put it in the this is one of the big advantages of having a big electrode in a small animal. You put a single electrode in here. So that's visual cortex, that's hippocampus, that's LGN. And over here, what you see is activity. This is cortex. This is CA1, dental gyrus. This is LGN, and this is another VPM, thalamic nucleus. Here you see the spike band voltage, see the units, you see the gamma power. You can see single unit evoked response because it's yoked to a visual input. So of course, hippocampus, dental gyrus doesn't have a visual evoked response, although they have a very high baseline rate. It's really interesting here. I hadn't really realized the close relationship, right? So here you see these are the CE1 pyramidal cell. They always fire in dental gyrus, but of course they're not related to vision, to visual input. Layer 4 is, and LGN is, and that's the local field potential. So it's really beautiful. I think this is going to be a cool tool. And together with Ken Harris, who's worked on using unsupervised learning machines, really seem to have solved the problem of spike discrimination. So I think that's going to open up an additional window that we use to do 2 photon at 30 hertz in horizontal, and then supplemented by doing these sort of columnar and subthalamic, subcortical sampling at 30,000 hertz. So we get very high frequency information, very precise timing information, without, of course, having access to genetic identifiable information. Right here, you don't know the identity of the neurons except they are in CA1 or in layer 2, 3, or layer 4 of cortex. Isn't that cool? All right, so I'm basically done. So our plan is now over the next several years to integrate, the wide field imaging with EFIS and with 2 photon, which is really a very demanding job. I mean, in principle, it's easy. In practice, it's enormous difficult to try to get, because we really want the database where you can click on it and you see, okay, here's 2 photon, and now the same animal, same trial, here's the EFIS, and here, by the way, is how it fits into the entire brain as the animal does its task. And then, yeah, we're doing modeling of a variety at different scales, from biophysical modeling to very abstract modeling of this activity, right? We want to describe this activity, not in the abstract, but in the concrete. We want to be able to replicate this activity based on our connectivity. We know these neurons are interconnected within V1 and to other areas. We know the electrophysiology under slice condition from our IVSCC. We have the observed behavior, so can we combine all of that and actually replicate the data? Yeah, and then we add a separate source of information is we have 2 different large sort of setup. One is with Stephen Smith using array tomography, where we know something about the identity using antibodies that we can wash off multiple times about the synapses and neurons. And then under Clay Reed, which is part of the IARPA, we're doing a very detailed reconstruction over the next several years of 1 by 1 millimeter of the same cortical tissue of animals that we have imaged in our own labs. So we can connect so we'll be able to know, if we zoom in, we'll be able to know the identity, the functional identity of some of these neurons. We can say that's a vertical selective cell, and that's a cell that responded very strongly to Touch of Evil, that one scene where they kiss, and here are the individual synapses. And so these are different antibodies to different pre- and postsynaptic component on excitatory and inhibitory cells. And yeah, that's our team. So the challenges we have, there are significant challenges. Of course, A is a challenge that every scientist has, is just setting up state-of-the-art instruments and getting it all to work, etc. The additional challenge we have that consumes much of my time is sort of because there's this team aspect to it, right? So we are a large team. So we just had a PNAS paper, there are 150 authors on it. Physics, it's getting to where physics is. And so if you go so I went to LIGO and I went to CERN. And if you talk to people in CERN, what's their match? So CERN, the Higgs boson, there were 2 big experiments. You probably know CMS and ATLAS. Each of them had 3,000 people, scientists, which includes staff from CERN, but mainly they were university faculty, university postdocs, and university students. And they have very detailed rules about everybody gets points, and if you have a certain number of points, you get to be an author, and who can speak. So academics are capable of doing large-scale projects together. It just has to, we just have to design things differently. How do you reward people? If you come to the Allen, clearly you can't come because you want to have that first author paper, because on a paper of 150 authors, there are only going to be 1 or maybe 3 start first author, right? So you have to think about other ways of rewarding, the sense of participating in a unique mission, the sense of being in a team. So being in a team implies you really have to trust other people, and there's sort of a whole dynamic around that, and it makes it more challenging than having small people, postdoc and a faculty. It also makes it very fun, because after all, we're a very social species, and we like working. I mean, it's a human genius. We can work together in pursuing distant goals together with many people. All right, thank you very much.